Hello, my name is Shahania Talak. Welcome to the Aphona Lab. Today we will be introducing polymerase chain reaction, or PCR for short. My colleague here, Morgan Chandler, will be performing the procedure. This procedure will require 1 microliter of the DNA template, 1 microliter of forward primer, 1 microliter of reverse primer, 22 microliters of ultrapure water. The water we will be using is between 17.5 and 17.8 mega ohms centimeter, and 25 microliters of Taq polymerase. We are using 2x MyTaq mix from Bioline. The equipment required for this procedure are 0.65 milliliter Eppendorf tubes, PCR tubes, a thermocycler, a microcentrifuge, and a vortex. To start off, the Eppendorf tubes are labeled. Here we have samples 1, 2, 3, 4, 5, 6, 7, and 8 to be amplified. The samples and reagents need to be thawed. Here, the samples are being warmed up in Morgan's hand. The reagents go on ice, and the samples get vortexed, centrifuged, and put on ice as well. 25 microliters of MyTaq mix is added to each sample tube. Followed by 22 microliters of double deionized water. One microliter of each forward primer, reverse primer, and corresponding template are added to each sample tube. After adding the forward and reverse primers and the template, use a pipette to pipette up and down to mix the sample. Remember to never vortex enzymes as they can be denatured. Pipette up and down slowly for about 10 times, taking care not to cause any bubbles. With those solutions prepared, we need corresponding PCR tubes prepared. 8 PCR tubes will be labeled, and 50 microliters of each sample are put into corresponding PCR tubes. The PCR tubes are placed into the thermocycler, and the following parameters are used: 95 degrees Celsius for 2 minutes, 94 degrees Celsius for 1.5 minutes, 55 degrees Celsius for 1.5 minutes, 72 degrees Celsius for 1.5 minutes. This is run for 30 cycles and kept at 4 degrees Celsius indefinitely. This process should take about 3.5 hours. When it is done, the DNA can be purified or stored at 4 degrees Celsius until purification. Following this procedure, 1 microliter of each template DNA was amplified into 50 microliters of unpurified DNA. The link to the purification video can either be found on the screen or in the description below. Thank you for watching, and thanks to Morgan for joining us. This has been the Aphona Lab.